You are welcome to my channel. From time to time, lecture videos on topics in microbiology will be posted here. The topic to be discussed today is phagocytosis. Phagocytosis is an important innate immune response against invading microorganisms and foreign bodies. What is phagocytosis? Phagocytosis is a multi-step process in which pathogens are recognized, ingested, and destroyed by phagocytes, which are specific immune cells in the body. There are steps of phagocytosis, and these include chemotaxis of phagocytes, migration of phagocytes, recognition of pathogen, ingestion of pathogen, killing of pathogen, and exocytosis of microbial debris. There are mainly three types of phagocytes. These include macrophages, which could be fixed or free. The fixed macrophages include the Kupffer cells in the liver, the alveolar macrophages in the lungs. Also have the polymorphonuclear neutrophils and then the dendritic cells. The first two steps of phagocytosis will be taken together: chemotaxis and migration of phagocytes. During an infection, the level of phagocytes increased. There is the release of a chemoattractant signal informing the phagocytes to migrate to the site of infection. And this means that the phagocytes should migrate quickly to the site of infection. Polymorphonuclear neutrophils attach to the endothelial cell surface by means of adhesion molecules. On arriving at the site of infection, the phagocytes have to recognize the pathogens. On the cell surface of the pathogen are pathogen-associated molecular patterns, which have to be recognized by pattern recognition receptors on the phagocytes. Example of pattern recognition receptor is the toll-like receptor. Example of pathogen-associated molecular pattern on the pathogen is bacterial bacterial lipopolysaccharide, LPS, which is recognized by toll-like receptor 4 present on the cell membrane of phagocyte. The next step is ingestion of the pathogen. The phagocyte extends its cell membrane around the pathogen. It's like you are welcome in a warm embrace, and the pathogen is engulfed into the phagocyte. A membrane-bound body known as phagosome is formed, and the interior of the phagosome becomes acidified. The content of the lysosomal granules need to be poured into the phagosome. But first of all, the lysosomal granule will fuse with the phagosome to form a new structure known as phagolysosome. The low pH in the phagolysosome leads to a release of degradative enzymes, including lysosome. The lysosome will destroy the bacterial cell wall, specifically the peptidoglycan. Then defensins, which are toxic peptides, create holes or channels in the bacterial membranes. The membrane is charged with the responsibility of maintaining the integrity of the cell. So if holes are created or holes are made, it means that nutrients or substances will move out of the cell, of the pathogen in this case, without control. That will be the beginning of the death of the pathogen. Following the creation of holes in the bacterial membranes by defensins, oxidative burst is the next stage, which is caused by the release of reactive oxygen intermediates. These reactive oxygen intermediates include superoxide radical, hypochlorous acid, singlet oxygen, and hydrogen peroxide. When the pathogen is fully destroyed or degraded, the microbial debris that is left is removed from the phagocyte by a process known as exocytosis.